Today, we are on site at the Fira Gran Via in Barcelona, Spain at the 2019 ESMO Congress. We'll be recapping some of the top news presented each day during the meeting, and soon we'll speak with Dr. Tony Choueiri on some exciting studies being presented in renal cell carcinoma and a couple other genitourinary cancer abstracts, as well as Dr. Omid Hamid on some pivotal melanoma trials. Welcome to OncLive News Network. I'm Gina Columbus. In hepatocellular carcinoma, results of 2 clinical trials show that patients with unresectable disease obtained clinically meaningful responses and had a statistically significant improvement in progression-free survival with the combination of atezolizumab and bevacizumab. More than 1/3 of patients attained objective responses in an uncontrolled evaluation of the combination, 3/4 of which were ongoing with a median follow-up exceeding 1 year. A randomized trial showed that the combination reduced the risk of disease progression or death by 45% versus atezolizumab monotherapy. Collectively, the studies provided a clear signal to proceed to phase 3 evaluation, but experts noted that there are several questions with the data that require answers. More than 1/3 of patients with previously treated locally advanced or metastatic cholangiocarcinoma with an FGFR2 rearrangement or fusion had durable objective responses to treatment with pemigatinib, a selective oral inhibitor of FGFR1, 2, and 3. The findings from the open-label, single-arm phase 2 FIGHT 202 trial showed an objective response rate of 35.5% in 107 patients with FGFR2 fusions and rearrangements, along with a median duration of response of 7.5 months. No responses observed in the 20 patients enrolled with other FGF and FGFR genetic alterations in another cohort, or in 18 patients with no FGF and FGFR alterations. In the first cohort, the higher ORR translated into a longer median progression-free survival of 6.9 months versus 2.1 months in the second cohort and 1.7 months in the third cohort. Overall survival data were immature at the time of the March 22, 2019 data cutoff. However, with a median duration of follow-up of 15.4 months and a median duration of treatment of 7.2 months, the median OS was 21.1 months in the cohort with FGFR2 fusions and rearrangements. In non-small cell lung cancer, an interim survival analysis of the phase 3 IMpower 110 study showed that single-agent atezolizumab improved overall survival versus platinum-based chemotherapy as a first-line treatment of patients with wild-type disease with at least 50% expression of PD-L1 on tumor cells or at least 10% expression on tumor-infiltrating immune cells. At a median follow-up of 15.7 months, the median OS was 20.2 months in patients randomized to atezolizumab versus 13.1 months in those who had platinum-based chemotherapy. The OS testing boundary was not crossed in the TC23 or IC23 wild-type population, so OS was not formally tested in this population, as well as in the TC123 and IC123 populations. Updated findings of the phase 3 Spartan trial showed that apalutamide in combination with androgen deprivation therapy demonstrated a 25% reduction in the risk of death compared with placebo and ADT in patients with non-metastatic castration-resistant prostate cancer. At a median 4 months of follow-up, results showed that the median overall survival was not reached in either arm, favoring apalutamide with a p-value of 0.0197. However, due to a pre-specified p-value boundary of 0.0121, these data were not found to be statistically significant. The data from the analysis also favored apalutamide regarding time to chemotherapy and PFS too. Finally, results of the randomized phase 2 Baracco study show that the combination of cediranib and olaparib is effective in heavily pretreated patients with platinum-resistant ovarian cancer, with an advantage of an oral administration and good tolerability. Moreover, the continuous schedule of cediranib and olaparib showed a promising trend towards improved progression-free survival compared with weekly paclitaxel, especially in the BRCA wild-type population. We are here with Dr. Tony Choueiri to discuss some of the wonderful genitourinary cancer abstracts that are being presented this year at the 2019 ESMO Congress. Dr. Choueiri is the director of the Lank Center for Genitourinary Oncology, director of the Kidney Cancer Center, senior physician at Dana-Farber Cancer Institute, and the Jerome and Nancy Kohlberg Chair and Professor of Medicine at Harvard Medical School. Hi, Dr. Choueiri. We're so happy to have you here today.
Thank you, Gina.
It's been a really great start to the ESMO Congress so far this year. There's been a lot of very interesting RCC abstracts. Can you first talk about the data we saw this morning of the Entrada trial with telaglenastat with everolimus in advanced metastatic RCC? So could you walk us through that trial and the data?
Yeah, that's an interesting concept, an interesting drug, and we do need actually new target in renal cell cancer beside VEGFR, mTOR, and the PD-1, PD-L1, and CTLA-4 inhibitors. And this target is related to the glutamine metabolism, and we know in cancer, we know now for over 100 years, the shift that occur in cancer metabolism, so, and the dependence on glutamine as well as other processes. So this is a glutaminase inhibitor with interesting preclinical data to back targeting this pathway in cancer. And as a proof of concept, this trial looking at the combination of this drug, the glutaminase inhibitor with everolimus versus everolimus alone as a control arm. As a proof of principle, that trial is positive, and the authors concluded that the combination is superior. I agree with that. However, one of the things that this is a very small trial, and the control arm everolimus didn't do well compared to what we and others reported on. There were no responses practically, very few, less than 5%, and the progression-free survival with everolimus was on the low side. Nevertheless, I think this is a proof of concept. The combination was tolerated. And what's interesting is that there is a much larger study with cabozantinib as backbone called Cantata. So we're looking forward to this study results.
Oh, all right, that should be a very interesting one.
Yeah.
So how about the final results of the TiNivo trial, in which tivozanib was combined with nivolumab in patients with metastatic RCC? What's exciting about these data?
Yeah, absolutely. Another very interesting combination in line with combining a PD-1 inhibitor with VEGF-TKI. Tivozanib is one of those very selected VEGFR inhibitors, and recently showed superiority over sorafenib in the refractory setting for progression-free survival response rate. And just recently, actually, the hazard ratio for overall survival, something that plagued this drug for a long time, became less than 1. In this study that presented at ESMO, the combination resulted in good response rate, over 50% combination, which you expect less with each drug alone. And the progression-free survival, despite this was small study, 25 patient, I believe, showed was more than 18 months with tolerable side effects. So hopefully this will be taken to the next stage.
Great. And with tivozanib, we recently saw an updated OS analysis. Can you kind of walk us through the history of this drug and its interesting timeline it's had so far?
Yeah, very interesting history. Again, one of the most potent VEGFR and clean VEGFR inhibitor, initially developed in the frontline setting against sorafenib, and despite winning on progression-free survival with a favorable safety profile, the hazard ratio was for overall survival was more than 1. And this was attributed in large part to the fact that patients on the sorafenib arm were allowed to cross over to tivozanib and receive tivozanib. So there was an imbalance with one arm, if you want, receiving 2 drugs and another arm receiving 1 drug. With a lot of patients actually accrued outside the United States with less options at that time. So this drug didn't get approved by the FDA, went to an ODAC and didn't get approved. So the following study, the TiVo-3, came, and tivozanib against sorafenib, but in a refractory setting in patients that received prior VEGF-TKI, and a large number of patients received prior PD-1 inhibitors. And again, tivozanib did win over sorafenib in terms of response rate, in terms of PFS, primary endpoint. But again, the overall survival hazard ratio initially was a bit over 1. An updated result showed, with more events coming in, more maturity of the data, that the overall survival was 0.99, less than 1. So hopefully there could be renewed interest in this drug coming. So we'll wait to see what the FDA will decide, how the path with this drug will be. And now we have data, very compelling data in combination with nivolumab.
Very good. Now, you're also on a couple of abstracts this year as well. Could you talk a little bit about the biomarker and efficacy analysis from Javelin Renal 101?
Yeah, so So Javelin Renal 101 was a study that established the combination of the PD-L1 inhibitor avelumab plus the TKI axitinib as superior combination compared to sunitinib, and led to the approval by the FDA. And in this two sub-studies that we presented, we look at patient with or without prior nephrectomy, and we looked at patient with or without prior sarcomatoid component in their tumor, which we know these are aggressive patients. And the combination was found to be superior irrespective. Something that I'll be presenting is the sarcomatoid histology, which we know it's not a proper histology. These are features present with every present even in clear cell RCC, and they're anywhere between 10 to 15%. And these patients historically were poor responders to VEGF TKI or even to mTOR inhibitors. But now there is renewed interest because we're seeing responses and we're seeing enhanced activity with checkpoint blocker compared to TKI. And there is a lot of biologic rationale. Increased PD-L1, increased CD8. There is also a immunosuppressive profile that these tumors have underlying, but checkpoint blockers work better, and I think there should be standard in this histology. Now, what type of regimen remains to be determined, but we know now that these patients with nivolumab and ipilimumab benefit much more than sunitinib. Same thing with pembrolizumab, axitinib. We showed now with avelumab and axitinib, atezolizumab and bevacizumab. So definitely, I would say frontline option with a patient with sarcomatoid histology should have a checkpoint blocker.
All right, thank you. So what about other abstracts being presented in RCC this year? What else are you excited about?
So one area I'm really interested about is always totally novel targets. So one target of particular is HIF2. So this was kind of an Achilles heel that was almost impossible to target because it's a transcription factor. This is really from a chemical biology and organic chemistry very hard, to target till investigators and researchers not long time ago found a compound that can actually inhibit HIF2. And this was presented, actually the first generation was presented last year, and this year we presented the result from the second generation, a drug called PT2977, which is a HIF2 inhibitor that recently was acquired from Peloton by Merck. And Dr. Eric Jonash and myself will present the data showing in a single agent activity over 20% in a very, very highly refractory population. So those are populations that treated with VEGF TKI, PD-1, median number of prior line of therapy 3. Response rate 24%, PFS 11 months. With an interesting enough, because when you start talking about a new target, you want to see how you combine these drugs. So you want to look at their toxicity. And actually, with the HIF2 inhibitor, there was some anemia, some hypoxia, but we didn't see the hypertension, the diarrhea, these VEGF class effect, a significant fatigue. So it seems to be quite well tolerated, hopefully promising for future combination.
Great. So moving away from RCC for a little bit, and let's go into some of the bladder cancer data that's being presented. So one that's eagerly anticipated is the IMvigor 130 trial. What do you think we could learn from the data when they're presented here?
Yeah, I think, we know that chemotherapy work in urothelial cancer and checkpoint blocker work. So we long anticipated combining chemotherapy with checkpoint blocker here, gemcis or carbo plus atezolizumab. So we have to see again the magnitude of the benefit. We would love to see something more so about overall survival, and would love to see something prominent about overall survival. The results in the lung cancer are there, and in non-small cell lung cancer with pembrolizumab, and it's quite, impressive. So I think we want to learn two things. We want to learn what is the magnitude of the benefit for PFS, for OS, for response rate, ultimately for OS. And second, if the magnitude of benefit clinically is not that high, if there is subgroup, and I automatically think about those patients with IC 2 and 3, with significant PD-L1 expression, are they the patient we should target with the combination? So quite exciting, and I'm happy that this will be presented at this meeting.
And we've seen some interesting novel targeted therapies being explored too in urothelial cancer. So there's some data with enfortumab that's going to be presented. We saw data this morning with sacituzumab govitecan. What are your thoughts on some of these more novel agents being explored?
Absolutely, and you say it better. I call them EV and SC. So antibody drug conjugate in bladder cancer are here to stay. So you just have the right drug with the right antibody with the right delivery system, and then you can see results. And both antibody drug conjugate are promising. EV is well ahead in its development, and we saw a trial led by Dr. Holmes that combined EV with pembrolizumab in cis-ineligible patients with quite an impressive response rate, 71% and over 90% tumor shrinkage. And seems that the combination could be tolerated. So one wonders if this combination is going to take over, let's say, pembrolizumab and cytotoxic chemotherapy. So that's quite interesting. I mean, the other combination, with SC, use a payload with irinotecan-like SN38, and also showed very interesting response rate with, as expected, hematologic toxicities, and should be moving forward along the same line. It will be interesting in the future, in my opinion, to see how do patients that progressed on EV do with SN38 payload and vice versa. And this remains to be seen.
Dr. Choueiri, thank you so much for being here today and sharing all of this insight with us. We really appreciate it.
Thank you. Thank you for having me.
Of course. We are here with Dr. Omid Hamid, who is the Chief of Translational Research and Immuno-Oncology at the Angeles Clinic and Research Institute. And he's here today to talk with us about some really exciting melanoma research that's being presented here at the meeting. Hi, Dr. Hamid, thank you so much for being here.
Hi, thanks for having me here.
Oh, sure. So let's kick things off with a reflection of some of the melanoma data that's being presented here at ESMO. So for example, we saw the 2-year primary results of TVEC in the neoadjuvant setting for select patients with melanoma. So what's the significance of these data?
The significance is that here we have something that's approved for locally advanced melanoma, which is easy to give. And what they're showing here is benefit prior to a surgical procedure. And the reason for this is hopefully improving relapse-free and overall survival. Now the data presented today is real early, it's at 2 years, but the trial was done at great institutions with surgical oncologists who have the experience. And I think the most significant portion is that they showed by just giving these couple of doses before you do the full surgical procedure, you could increase the T cell infiltration, you could increase the peripheral T cells that are activated. And that's an indication of where we can go in giving these patients who will ultimately then move on to adjuvant therapy or not need other adjuvant therapy a boost. Additionally, at this meeting, they are presenting more data with TVEC in combination with anti-CTLA-4 therapy, showing high response rates and durable benefit. So as we see here, a drug that's approved for locally advanced unresectable melanoma, trying to find other places where it can be helpful immunologically and give patients benefit.
Wow, very interesting. Okay. So the IMMUNIT trial is looking at single-agent nivolumab or in combination with ipilimumab as an adjuvant regimen in stage 4 melanoma with no evidence of disease. So could you share your thoughts on this trial and the data?
Right, so this is data presented by Dirk Schandorf and others in relation to stage 4 disease being treated with either single-agent nivolumab or combination nivolumab and ipilimumab. What it's showing here is benefit over doing nothing. What it's also showing is an indication that this can be done and be safe and tolerable. Additionally, what we saw here is that patients with brain metastases that were resected were given these therapies, and in these subsets had a survival that is better than what we historically have seen with patients with brain metastases. So as this data matures and the adjuvant data from 238, which is ipilimumab versus nivolumab, mature, I think it'll come together while we await Jeff Weber's 915 trial, which is adjuvant nivolumab with low-dose ipilimumab, and we can kind of get an idea about combinations in stage 4, a resected disease, stage 3 disease, and this builds our groundwork to look at other adjuvant therapies that are coming out, which are combinations. Right now there's a personalized vaccine plus anti-PD-1 therapy that's out there. So the beginnings of understanding how we can treat patients, whether early is better than late, and in understanding that the morbidity is low with this type of therapy.
That's a very nice segue into the 238 data. Can you share, now this is a little bit more longer term data with 3-year findings. What are we seeing now at this year's meeting?
So at this year's meeting, Jeff Weber updated the 238 data, which is a randomized double-blinded trial of ipilimumab versus nivolumab in stage 3B and above, including resected stage 4 patients. So a great trial which has brought anti-PD-1 into standard adjuvant therapy. As he's updated the data, what we've seen is that the benefit still persists, that the difference between anti-CTLA-4 and anti-PD-1 in relapse-free survival maintained through the many years. Additionally, we're seeing a plateau now, and that's beneficial. But the strongest data comes from some of the understanding about indicators of benefit. And I just like to focus on high tumor mutational burden and interferon gamma signature positive patients, where those patients who had both seem to have the greatest benefit. Now, we don't know if this is a prognostic marker or a predictive marker, but what this is doing, along with the data Georgina Long has presented with dabrafenib, trametinib, and the other data with nivolumab in the adjuvant setting, is giving us some predictive markers to understand which subset is going to benefit the most, and is setting a standard which should be part of how every other phase 1 trial moves forward, and allows us to kind of tweak how we treat patients in the adjuvant setting, stage 3 and stage 4.
Those are some great points. Now, this year we have 5-year outcomes of CheckMate 067 of nivolumab and ipilimumab in advanced disease. So what are these data showing, and what have we learned about this combination over the last 5 years now?
So CheckMate 67 being presented, and you'll remember that that is a ipilimumab alone versus nivolumab versus ipilimumab and nivolumab given in the classic fashion of ipilimumab at 3 mg per kilogram, and in looking at it in stage 3 unresectable and stage 4. This is again showing 5-year survivals greater than 50%. So we're starting to nudge up survivals. We're showing the benefit of ipilimumab, nivolumab, and single agent nivolumab over ipilimumab. So placing anti-PD-1 therapy, either single agent or combination, as the first line therapy for anyone with unresectable or metastatic melanoma. It is not saying that either one of these PD-1 combinations is better than the other, but it is giving us long-term data on which to base endpoints for future trials. The understanding that we should be moving forward with PD-1, and then showing us what we should be taking back to our patients in clinic, and saying that in just a handful of years we're taken the 5-year survival of metastatic melanoma from under 10% to above 50%. So that gives hope. And let's not forget that a lot, if not all of this immuno-oncology data with checkpoint inhibition has begun in melanoma, and the same bright future can be translated into other solid tumors, whether it's in the metastatic or unresectable care of patients or in the adjuvant or neoadjuvant.
Good, thank you. So aside from some of these other data we talked about, what are some research that you're involved in that's being presented this year that you'd like to share?
So as we go and get through the meat of the data that's being presented in melanoma, I just want to come back and say that what this has shown is, number one, clinical trials are the way forward. They're the future in improving outcome. That we still have a lot of work to do. 50% 5-year survival is not good for the other 50% of my patients, and that we should be moving forward. We are doing more work in understanding how to take care of the refractory population, whether it's with manipulation of the microbiome or other targets in melanoma. There's data on microbiome here that's interesting. Predictive and prognostic markers are key here, whether it's circulating tumor cells or initial gene signatures, interferon gamma, or tumor mutational burden. And then as we move forward in the developmental therapeutics portion, how we can do better with personalized vaccines, with microbiome, with combinations that are not only checkpoint, but are looking at other targets like VEGF. So that's the future. We are also at a time where moving forward with immunotherapy means the addition of adoptive T-cell therapies into our armamentarium, and those are becoming more and more available for our patients through trials like the IOVANCE trials. The idea of oncolytics and injectables, whether it's telemedine leheparebec or IL-12 or TLR9 or others, and then CAR T therapies, now not for your liquid tumors but for your solid tumors, and trials that are opening not just for melanoma but triple negative breast cancer, pancreatic cancer, ovarian cancer, melanoma, etc. So we are embarking on the next phase of our trials, having a great base for adjuvant, neoadjuvant, and metastatic based on our PD-1 and anti-CTLA-4 therapies.
That's great. It all sounds like there's so much on the horizon to really be excited about.
Right, and I think myself and my colleagues are going to have a good time, learn a lot, and head out of ESMO back to our clinics with a lot of work left to do.
Great. Thank you so much, Dr. Hamid. We really appreciate you being here
today.
Thank
you for the
sharing
opportunity. Appreciate it.
Thank you. That's all for today. Stay tuned for tomorrow's OncLive News Network On Location, where we will sit down with Dr. David Spiegel on lung cancer studies and Dr. Celestia Hagano on some pivotal prostate cancer data. Thank you for watching OncLive News Network. I'm Gina Columbus.